Hi, this is Michael Melman. Now we're going to talk about reproductive embryology, which can definitely be one of the more challenging aspects of the book conceptually. Fortunately, the USMLE doesn't require that you know an exceedingly broad range of topics, but rather a collection of high-yield facts and relationships. So with those in mind, let's get started. So the USMLE will occasionally assess a few genes that are responsible for embryological development. One of them is the Sonic the Hedgehog gene. So imagine that this is your trunk here, and then this is a developing limb bud here. And as this limb grows, whether it's an arm or a leg, it's going to have different appearance and different function along the anterior-posterior aspect. Okay? So the Sonic the Hedgehog gene is responsible for that. The USMLE, most importantly though, it will actually want you to know that the Sonic the Hedgehog gene, mutations in it are responsible for holoprosencephaly. Okay, holoprosencephaly, where during development you have your forebrain that will lead to two hemispheres. Okay, so your forebrain is also known as your prosencephalon, which the prosencephalon is the telencephalon and diencephalon. So your telencephalon becomes your cerebral hemispheres and lateral ventricles. Now you have two cerebral hemispheres, right? And you have two lateral ventricles. But if you have a mutation in this gene, you have holoprosencephaly, you will only have one cerebral hemisphere, okay? One lateral ventricle. And the same thing goes for the diencephalon, which will be your thalamus, your third ventricle, and your cerebellum, okay? So you won't have bilateral patterning, but instead just a non-division. Okay, this holoprosencephaly. Sonic the Hedgehog mutation is one cause of that. The USMLE likes to test that if it does show up. And I'd say another high-yield piece of information is that if the USMLE is going to test you on holoprosencephaly, you should remember three things. One of them being Sonic the Hedgehog gene mutation, as we've discussed. The second would be Patau syndrome, which is trisomy 13. Okay, these children die usually before the age of 1 if they do come out alive, but holoprosencephaly is a characteristic of that syndrome. And so holoprosencephaly can be caused by, among other things, a mutation in Sonic the Hedgehog gene, Patau syndrome, and severe fetal alcohol syndrome. Okay. In contrast, the Wnt family of genes is expressed in the apical ectodermal ridge, which is the most distal part of a developing limb. So whereas we had the Sonic the Hedgehog gene, which was responsible for patterning along the base of a limb, the Wnt7 gene is responsible for patterning along the distal aspect of a growing limb. And you can remember that because the Wnt7 gene, 7-year-olds are still growing, so their limbs are becoming more distal. The fibroblast growth factor genes also have a variety of roles in embryos and in adults, but on the step 1 you will most likely see them in the context of limb development. Like the Wnt genes, they are produced at the apical ectodermal ridge, where they stimulate mitosis of underlying mesoderm, providing for lengthening of the limbs. One of the favorite genes that the USMLE will assess is the FGFR3 gene, the fibroblast growth factor receptor 3 gene, which you just got to know that if you have a mutation in that gene, that's responsible for achondroplasia, which is a shortening of the limbs despite having a normal trunk and head size. Okay? So about 80 to 85% of the time, that's a de novo mutation. 15 to 20% of the time, that is autosomal dominant. Achondroplasia associated with growth mutation in FGFR3 gene. And as you can see here, the fibroblast growth factor genes do have a role in limb development. The homeobox gene, also known as HOX, is important in the segmental organization of an embryo in the craniocaudal direction. Okay, so different combinations of the HOX genes cause different segments to have distinct fates. So for example, the HOXD13 gene is expressed in the metacarpals and phalanges, but not in the carpals, for instance, where you can see that 12 is expressed. So incorrect expression or mutations in HOX genes can result in major changes in development or localization of appendages. So what you got to know that's very high yield for the homeobox gene is that mutations will cause appendages in the wrong locations or polysyndactyly. So syndactyly would be fusion of the digits, so fingers or toes, whereas polydactyly would be multiple, such as 6 instead of 5 fingers or toes. Okay? So polysyndactyly and appendages in the wrong locations on step 1 equals mutation in homeobox gene. Okay? Whereas holoprosencephaly, mutation in Sonic the Hedgehog gene, Patau syndrome, or fetal alcohol syndrome, mutation in FGFR3 gene, achondroplasia. All right? And apart from needing to know that the homeobox mutation can result in appendages appearing in the wrong locations or polysyndactyly, the USMLE also really wants you to know that the HOX gene codes for DNA binding transcription regulators. Okay? DNA binding transcription regulators. And interestingly, this does make sense because lack of regulation of gene transcription in the event of a HOX gene mutation could conceivably result in improper gene expression and limbs appearing in the wrong locations. For the flash quiz, out of the following, Sonic the Hedgehog gene, Wnt7 gene, FGF gene, or HOX gene, mutations in which are associated with holoprosencephaly? That would be the Sonic the Hedgehog gene. Now let's discuss the general timeline of development. This information can be useful in many cases, such as knowing when teratogens are most dangerous and knowing when congenital defects can be tested. Let's start from the beginning with fertilization. First, in the fallopian tube, a sperm finds a secondary oocyte that has its cell cycle arrested in which phase of meiosis? That would be metaphase of meiosis 2. Okay, so the oocyte, the secondary oocyte that's released by the ovary at ovulation is halted in metaphase of meiosis 2 until it is fertilized by a sperm. So the sperm undergoes an acrosomal reaction to release enzymes which degrade the zona pellucida of the oocyte, allowing it to pass through so that the gametes can fuse. It should be pointed out that the acrosome of the spermatozoon is derived from the Golgi apparatus and the flagellum from centrioles. In response to membrane fusion, the oocyte releases enzymes from its cortical granules, causing the zona pellucida to become impenetrable to additional sperm. The oocyte then finishes meiosis 2 and releases a polar body. The nuclear membranes of the oocyte and sperm are then degraded and reform around the diploid genome. At this point, it is a single-celled zygote. In week 1, as the zygote travels through the fallopian tube, it divides into a mass of progressively smaller daughter cells. And by about day 3, what has the zygote become? It has become a morula, which is a compact ball of about 32 cells, by about the time it reaches the uterus. Next, it turns into a blastocyst in which the cells surround a central cavity called a blastocele. Between the 7th and 10th day, the embryo implants in the wall of the uterus. And human chorionic gonadotropin, or HCG, secretion begins shortly after. Since pregnancy tests detect HCG, it can first be detected by blood or urine tests between 8 to 14 days following fertilization. Do you know which structure prevents implantation before day 7? The zona pellucida, which is not shed from the embryo until the blastocyst stage. During the first week, the only differentiation that occurs is into the trophoblast, which will become the placenta, and the inner cell mass, which will become the embryo, amnion, and yolk sac. During week 2, the inner cell mass begins to differentiate into the precursors of different tissues. The inner cell mass divides into the bilaminar disc form, which is exactly what it sounds like, a disc composed of an epiblast layer and a hypoblast layer, with the epiblast located dorsally and the hypoblast ventrally. The epiblast is mostly associated with ectoderm, but is actually a precursor to all three germ layers, since it invaginates to form the primitive streak, which we'll talk about more later. And as you can see in the image here, two cavities form as well. The dorsal one is the amniotic cavity, and the ventral one is the yolk sac. During week 3, the process of gastrulation occurs. It is the process by which the bilaminar disc changes into a trilaminar disc, and those three resulting layers are what will go on to form the ectoderm, mesoderm, and endoderm. Week 3 is also when neurulation begins, formed by invagination of the neuroectoderm. The notochord, a rod of cells that runs along the craniocaudal axis, induces the formation of the neural plate by secreting morphogen sonic hedgehog, which we talked about earlier. This causes neuroectoderm to roll up like a tube, with the edges of these neural folds fusing together at the midline by week 4. The alar and basal plates are the dorsal and ventral parts of the neural tube respectively. The alar plate gives rise to a lot of the sensory parts of the nervous system, and the basal plate contributes to most of the motor components of the nervous system. Now, the neural tube formation begins in the neck region at the 4th somite and proceeds in both cephalic and caudal directions, with the cephalic being completed first. Do you remember which vitamin deficiency most commonly causes neural tube defects? If you said folate, you're right. Okay, and it is also really high yield that heart development and limb development both begin at week 4. You can remember this easily because the heart has 4 chambers and there are 4 limbs. So next, the cephalic end will dilate, forming the forebrain, midbrain, and hindbrain, while the rest of the neural tube forms the spinal cord. And as the neural folds elevate and join at the midline, cells at the edges of the lateral plates, or neural crests, differentiate to form neural crest cells. When they reach their final destinations, they differentiate into numerous cell types, including spinal ganglia, melanocytes, and support cells of the nervous system. So the USMLE wants you to be aware that the fetus is most susceptible to teratogens between 3 and 8 weeks. They'll ask you the time frame. Most students say 0 to 3 weeks. It's wrong. The answer is 3 to 8 weeks. If there is significant exposure to teratogens during 0 to 3 weeks, it'll result in the all or nothing phenomenon. There'll be a spontaneous abortion. The mom won't even know she was pregnant, or the fetus will generally heal to the point that there's no phenotypic problem. So 3 to 8 weeks, when, as we talked about before, we've got heart development and limb development. Okay, by 6 weeks, you could detect the heartbeat on ultrasound. 8 weeks, fetal movement. And if they ever ask you about male or female genitalia, it's by 10 weeks that they're first differentiated. By all means, in clinical practice, the gender cannot be identified until the 2nd trimester ultrasound, which is roughly 16 to 19 weeks. But in terms of embryological development, it's at 10 weeks that the male-female genitalia can first be distinguished. So for the flash quiz, when does the heart begin to beat? And as we discussed, because this is very high yield, it's 4 weeks. You can remember 4 chambers, 4 weeks.